Hello, and thank you for being here. My name is Edoardo Sarti, and I will present my talk titled Paralog Function Annotation with ProfileView. This work has been done in collaboration with Alessandra Carbone, Julien Henry, and Théo Lemoigne, all at the LCQB laboratory at Sorbonne University. The team led by Julien Henry is doing an extensive effort for determining the structure of all the 11 proteins participating in the Kelvin-Benson cycle in Chlamydomonas reinhardti, a unicellular algal model organism. The Kelvin-Benson cycle is one of the main chemical pathways present in all photosynthetic organisms. Its role is to fixate the carbon present in CO2 molecules into glyceride 3-phosphates or G3P molecules, which ultimately will be used to produce glucose. Contiguous proteins in the cycle share their ligand and thus are likely to interact. But there is strong evidence that there are also long-range interactions in the Kelvin cycle, such as the one between GAPDH and PRK, mediated by the small CP12 protein. Are there other interactions between non-contiguous proteins in the Kelvin-Benson cycle? A common approach to protein-protein interaction prediction is to detect possible coevolution signals between pairs of proteins. For doing this, we need to build a joint multiple sequence alignment starting from the MSAs of the two chosen proteins, and then to use one of the many state-of-the-art algorithms for finding clusters of co-evolving residues across the two joined parts of the alignment. For this task, we are using IBIS2, an accurate, iterative method able to detect groups of co-evolving residues even in case the correlation signal is only present in a subset of sequences. The IBIS2 method is also robust with respect to sequence conservation. You can find out more about our recent online implementation IBIS2 analyzer during the poster session. The MSAs of Kelvin-Benson cycle proteins present specific characteristics. They have the advantage of having many sequences, although highly conserved. On the other hand, due to the long evolutionary history of this pathway, each protein has several paralogs that share many common sequence features but play different roles inside the cell and do not contribute to the Kelvin cycle. This is a problem for our approach. If we incorrectly couple the sequences coming from the two joined MSAs, the coevolution signal will be completely lost. The problem is thus, for each species, how do we find the correct partner among the paralogous proteins? This task requires knowing the function of each protein, but functional annotation of paralogs is notoriously difficult, both with specific experimental assays and with large bioinformatics studies. Recently, a new method for function prediction called ProfileView has been published by the team of Alessandra Carbone at LCQB. The algorithm is based on the use of hidden Markov models and hierarchical clustering, and does not need any multiple sequence alignment. For building the library, ProfileView considers a set of seed sequences provided by the user, and for each one of them, it searches into a sequence database for hits with very high sequence identity. A hidden Markov model is thus built for each query sequence and added to a library whose scope is to store profile information about the seed sequences. When the library is built, the user enters the protein sequences that need to be functionally classified. At this point, a classical phylogenetic algorithm would consider their distance in the sequence space and build a phylogenetic tree with a hierarchical clustering method. Instead, ProfileView embeds the sequences in a representation space defined by the HMMs contained in the library. This space has one dimension for each HMM. When a sequence is aligned against an HMM, a likelihood is returned estimating the probability of that sequence to have been produced by the HMM, that is, to be similar to the group of sequences the HMM was trained on. Thus, each query sequence is aligned against all HMMs, and the resulting vectors of likelihoods is interpreted as the coordinates of the query sequence in representation space. Finally, hierarchical clustering is applied in order to obtain a dendrogram, which will be based on this functional space. ProfileView has reached excellent performances in predicting the function of homologous proteins, but has never been used for Paralog function annotation before. We thus have used a new protocol in order to maximize the signal. For each Calvin cycle protein, a list of pertinent PFAM domains is extracted. We start from the PFAM full multiple sequence alignment and on subsequent steps of clustering with MM6 for choosing around 1,500 sequences with which to build the model library. These are thus sequences relative to functionally important and conserved domains. Indeed, considering full proteins sequences might introduce unwanted noise. Also, there is no taxonomic selection on the sequences used for library construction. A remarkable fraction of them comes from eukaryotes and other non-photosynthetic organisms. This is found to facilitate ProfileView in its functional classification, as it provides a more consistent set of negative examples. Then, the maximal set of photosynthetic homologous sequences is entered in ProfileView in order to be classified. For each protein, we have manually curated the annotation of a set of sequences in order to see whether Calvin-Benson and non-Calvin-Benson paralogs are correctly classified in separate subtrees. Most of the available records come from Chlamydomonas reinhardti, Arabidopsis thaliana, Volvox carteri, Spinacia oleracea, and some species of Synechocystis. This is the result of ProfileView analysis on the fructose 1,6-biphosphate FBA, where we can clearly see the Calvin-Benson paralogs being clustered in a different subtree with respect to the non-Calvin-Benson ones. We obtain comparable results for 8 out of 11 proteins. Out of the remaining 3, the method does not show any separation in the case of the rubisco, which indeed has no paralogs. In the other 2 cases, ProfileView does differentiate between Calvin-Benson and non-Calvin-Benson proteins, but the data points happen to be in a very limited region of the tree, and further analyses are needed. With these results, we are now starting to address our original question, which implies the calculation and manual analysis of a large number of protein pairs. As a preliminary result, we started from the only known case of protein-protein interaction between the 2 Calvin-Benson proteins that do not share any common ligand: glyceride 3-phosphate dehydrogenase, GAPDH, and phosphoribullokinase, PRK. IBIS2 finds several coevolving clusters across the 2 proteins, but upon visual inspection they do not seem to form surfaces that could interact. It is only by considering the CP12 protein that we see how these clusters are functionally linked. Both are indeed found at the interaction sites between the 2 proteins and the mediator CP12, in agreement with the recent experimental evidence. In summary, Calvin-Benson proteins have multiple paralogs, and it's hard to guess which one would take part in the Calvin-Benson cycle. ProfileView correctly classifies the function in 8 out of 11 Calvin-Benson cycle protein families. Preliminary results show the functional classification can be used for protein-protein interaction prediction. Thank you for your attention.